Okay, enzyme. What is enzyme actually? So enzymes are protein molecule, as I said before. So these molecules it act as catalyze. Catalyze means speed up a reaction. So since the enzyme is found in the body or in a living thing, so we use the word biological catalyst. So this biological catalyst, which is the enzyme, can speed up the biochemical reaction. Biochemical is inside the chemical reaction in the living thing. So biochemical reaction without themselves being changed at the end of the process. So they will be the same after the reaction. So they won't change into anything. Okay, that means how it come after the reaction, it will be remain the same. Okay, so the second thing is these enzymes are actually tertiary proteins. So you can see up here, okay, as I mentioned just now, alright, it has this beta plated, which is secondary, and then it has this single chain, which is the primary. So then this primary and helix, alpha helix also there. So when you have primary and secondary combine, you will form your tertiary proteins. Okay, now why or where the enzyme is produced? So there are two types of enzyme here. Okay, so enzymes, okay, where first? Okay, not types. Okay, where? So enzyme is produced in the ribosome. Okay, ribosome are the one that produce. So as we have learned last time, in the previous chapter, this memory or this enzyme, the protein, is inside the DNA, alright, or inside our nucleus. So from the nucleus, they will send this short DNA, a sequence of DNA to the ribosome, so that the ribosome can translate it, okay, can translate it into the specific amino acids, or arrange the specific amino acid into the enzymes needed, or the enzyme that is need to produce. Okay, so why does this ribosome can be found? And in chapter 2 we have learned that we can see this ribosome either on top of the rough ER, okay, stick on the top, attach the rough ER, or is suspended in the cytoplasm, which is we call the free ribosome. So when it's on the rough ER, so when the ribosome get the message from the DNA, or we can say when the information is sent out, okay, from the nucleus to the ribosome will start to translate whatever message on the DNA, and then they will form the particular enzyme, particular proteins they need. Okay, so the particular protein then will shape, okay, into different shape, okay, or it will undergo primary structure, secondary structure, and then tertiary structure until it becomes the enzyme that is needed. Okay, so there are two groups of enzyme, or two types of enzyme. One we call it intracellular, another one is extracellular enzyme. So intra, the word in means inside, extra means outside. So what is outside, inside? So intracellular means it's formed inside the cell, of course, from the ribosome, and then it retained inside, okay, for the use of the cell. Okay, the cell need this enzyme, so it is formed inside the cell and then it keep inside the cell so that it can be used for the biochemical reaction. For extracellular enzyme, is formed in the cell. Of course, it's in the cell. Ribosome is inside the cell, but after that, it will be packed and secret out from the cell to do the function. Okay, so what we can see here, okay, the example, okay, the example are DNA polymerase, RNA polymerase, and ATP synthesis. These are the enzyme. These are the enzyme which is used inside, okay, DNA polymerase. So we can see that here from the name, this enzyme ASE, you see the word ASE here. So ASE here means this is an enzyme. So whenever you see the word ASE, it means this enzyme is an enzyme, the name for enzyme. So DNA polymerase means this is a enzyme that speed up DNA become the polymerization, okay, to form DNA. This one is for RNA, okay, this one is for ATP synthase, so an enzyme to help in synthesis of ATP. Okay, the name help us to know what an enzyme do. Okay, now intracellular, another point is occur in cytoplasm, occurs, okay, organelle or nucleus. Okay, so this is where it is used. Okay, so this one extracellular is produced in the cell and then is sent out, okay, to catalyze the reaction outside the cell. Example, digestive enzyme. So this is outside, which is in your mouth or in your stomach or in your small intestine. So those are extracellular enzyme which is produced by the cell and then sent out to respective organs. Okay, so mechanism of enzyme. So this is what I said. Okay, so beginning, if you have enzyme, this yellow one is enzyme. So this enzyme will have their active site. So active site is formed due to the tertiary structure. So the protein, the amino acid will bend and turn or twist into a specific site like this. Okay, every substrate will have different site or different active site. So one substrate, if the substrate is fit, okay, if the substrate the shape is the same as the active site, then it can bind and become ES, mean enzyme substrate complex. So when they are fixed together, they can fit in, so we call them the enzyme substrate complex. Okay, enzyme substrate complex. So substrate is the, for example, this is maltase. Maltase means maltose. Okay, the substrate is maltose, the sugar. So in order to break down maltose into glucose, you need maltose here, and then use maltase. So maltose plus maltase, it become the subcomplex. So the complex now, it will have the precise shapes called the active site. So this active site is the one. Okay, so after they bind, okay, the enzyme will fit directly into the active site. Is like a lock, the key into the lock. So we call this the mechanism, the lock and key hypothesis. There are many other hypotheses related to the mechanism of enzyme, but what we will see here is lock and key. Alright, if you go Google, you can see other things. Okay, now next. So this is due to the surface configuration, means the shapes of the enzyme, then it will form the specific site. So what is enzyme substrate complex? Okay, so when they bind, we call them the enzyme substrate complex. This is another drawing or another pictures here. So actually, when we say the shapes, is actually the bonding, how it form the bond and the ions there. So you will see the H and the N there, H and the N there. So when they form, okay, when they form actually the binding of the ions, okay, positive and negative ion. That one will be complicated a bit lah. So what we can say is just enzyme bind with the substrate, the substrate bind to the enzyme and become enzyme substrate complex. Now this is another picture showing how they split. Okay, so if you have one substrate, just now I'm using the maltose as my example. Maltase is the enzyme. So you see they combine. So the green one is your substrate, the gray one is the enzyme. So after they become the complex, the reaction happen. So if it's a catabolism means it break. Okay, you see the product become two, from one become, okay, it become two, from one here become two here. Okay, so it breaks, so catabolism. So but you see the enzyme will remain the same. Okay, it will not break. Okay, I said you have other substances which is not maltose. Okay, maybe most of the time if it's not maltose and this is maltase, the shape will be different, so they cannot fit in. Okay, they cannot fit in, so it won't have this reaction. So that means only one enzyme can only catalyze one substrate, one type of substrate. Okay, either go like that, okay, from either splitting it, okay, hydrolyze, or combine it, because it still have the shape. So as long as if I have maltose, one is glucose, I join, I can still get back my maltose. Okay, this example, since this one is different shape, one is about half oval, this one is triangle, that means maybe this is not maltose lah. This may be sucrose. You have maltose, sucrose you will have glucose and fructose. So sucrose you have glucose and, sorry, this one I get from the internet. Okay, so you have glucose and then fructose. So if another time when you have glucose and fructose in the blood or in the cell, so it can also bind to the same maltase. We are using sucrase. Okay, sucrase. So it can use back the same enzyme sucrase to form back, okay, sucrose. Okay, so this is what it means. Okay, the enzyme can split or link. The same enzyme, the gray one, it can split. Either it go like that in that direction, or it come back this direction. Okay, either it's hydrolysis or condensation. Okay, so once it's formed, the product no longer fits, so it will escape or leave the enzyme. Okay, now, so just now we are talking about the mechanism. So now we will see generally what are the characteristics of the enzyme. So first thing, of course, is speed up the rate of chemical reaction by lower down the activation energy. Second, it will not destroy. You see, it only help or increase the speed. Okay, speed up. Okay, lower down the activation energy. So after the chemical reaction, the enzyme release from the attachment. So it is just released like that. Okay, it will not change or it will not destroy end of the catalyzed action. Okay, now enzyme catalyzed reaction are reversible. So as I said, okay, I have lactose using lactase. Okay, and then you have glucose and galactose. The same substance, galactose and glucose, can also use the same enzyme become lactose. So from lactose become the glucose and galactose. That one is hydrolysis. Go backward will be condensation, which we learned in the previous chapter. Okay, enzymes are highly specific. So what is meant by highly specific? So one catalyze only one kind of substrate because of the shape. Okay, so but it's reversible. Same substance and same enzyme is reversible, but you cannot use lactase to speed up the reaction of maltose. Okay, that's cannot. Okay, mean by specialized. So since it can be reused again, so it will needed. Okay, it was not used up. It can be reused, so it need in small quantity, so your body will not produce too much. Okay, no need to produce so much of enzyme, just enough because it can be reused. Okay, of course, the more we produce, we can have the activity or the biochemical reaction happen faster, but there is waste of energy. Okay, number 6. Okay, enzyme activity can be stopped or slowed down by an inhibitor. So this one is to help to control enzyme activity. Okay, for example, we will say the enzyme will not it will slow down and it can be reused. Okay, it will not destroy. So how to stop an activity? If it cannot be destroyed, you have to stop it somehow. Okay, how to stop the enzyme activity? If it cannot destroy, if it will keep on happening. Okay, which is not logic. Okay, so there are substances, or we call inhibitors, that can stop or slow down the enzyme. Of course, sometimes it's a good thing, sometimes it's not a good thing. Okay, some inhibitor which is a bad one. Bad one means it stop the activity which we need. Okay, then it's not good. But let's say it's a activity, it stop the activity that we have enough. Okay, then it's good lah. So it's not really a bad thing, but it's just a control or regulation. You see, a regulation of the enzyme activity. So another characteristic is it is sensitive to temperature. Yeah, so all the enzymes are actually very sensitive to temperature. That's why when you get fever, it's not cool. Okay, it's not good. Okay, because our enzyme in the body optimum temperature is around 37. Okay, all the enzyme that is produced by our body will have this temperature to work best. Okay, to work at best. So if the body temperature rises, okay, for example, you have fever, so the enzyme will become denatured. Okay, denatured means it will not be used anymore. Low temperature is still okay, you see, it's still functioning, but somehow if your temperature is beyond 40, 50, okay, the enzyme will totally change the bonding. Okay, just like chemicals, you go and burn. Okay, if you burn your chemical, the bond breaks. Same to your enzyme. So when it happen in our body, we call the enzyme is denatured. Okay, the enzyme will denature if the temperature is too high, and it is not reversible. After it denature, it cannot come back. Okay, it's the same goes to pH. So all the enzyme in our body, it responds to different pH. So if this is a pH scale, 1 to 14, okay, here is 10 only. So here is acidic, this is more to neutral, alkaline. So you see the graph for this is different, okay, different enzyme. So pepsin is a protein, okay, amylase, okay, in your mouth, okay, and trypsin is in the intestine. So different enzyme here will have different pH to work the best. Let's say I take pepsin in an acidic condition, it can work best, but from the acidic condition I bring into a neutral condition, okay, the pepsin won't work at all, okay, it will just stop. Similar goes to the salivary amylase. If I bring it into the stomach, it will just stop because it's too acidic. But good thing here, if I bring the if amylase is entering acidic, it will just stop only, okay, it will just stop. And then if I can reverse, if I can from acidic I can change it back to neutral, this amylase can work again, okay, can function again, okay, only at extreme pH lah, okay, they may denature, okay, but some or most of the enzyme, okay, if I return or what we call, we can bring back the pH into their pH, it still can function. Okay, number 9, the property number 9, it will remain unchanged lah, so it can be reused. So this is about the characteristics. Okay, the next part is about naming the enzyme. So as I said just now, so by looking at the name, I can actually know what is the substrate does that the enzyme act on. Okay, so you can see if my substrate is maltose, I will use maltase. Okay, if my substrate is lactose, I will use lactase. So this, the one that is come out is product. Okay, so if I have sucrase, okay, you can guess, yeah, the substrate will be sucrose. And then how, okay, this is how, okay, so this is example maltose. Okay, so the enzyme, I take away the OSE, I just replace it with ASE, so maltase. Okay, for lactose, okay, if the substrate is lactose, the enzyme will be, I take away the OSE, replace it with ASE. Now last one, sucrose, same thing. So whenever you see the word ASE, it is the name for the enzyme, except for a few earlier found enzyme, yeah, for example protein, you have pepsin just now, pepsin, trypsin, okay, this is actually enzyme for protein, which is found earlier, okay, earlier before this what we call the naming, this the rules of naming enzyme is being introduced. That's why they still use the old way of naming. Okay, so it's understood lah. Okay, pepsin is actually polypeptidase. Okay, trypsin is peptidase. Okay, so it has their own name in a naming, but we have used to the name like pepsin and trypsin, so we just go ahead lah, we just use back their normal name. Okay, so we will stop here for the name. Okay, after this I will have another video on the factors affecting the enzyme. Thank you.